Good morning. There's lots of great seats right down front. This is not church. Welcome again. And I want to introduce Dr. David Threadgill, who will introduce our first speaker, Dr. Bill Murphy. So.
Howdy, everyone. So, I've got a few comments I want to say, but I want to first off and say it's really a pleasure to introduce Bill Murphy today. Bill is a good friend and colleague here at Texas A&M. And before I get to my introduction to Bill, I want to sort of say a few words about Jim Womack and sort of my experience with Jim. When I actually finished my undergrad degree in the biology department here at A&M, I had no clue what I wanted to do. I'd taken a genetics course that I thought was real fascinating. And John Gold was my instructor, and he says, well, why don't you go out to the vet school and talk to this guy? We have a new faculty member out there that has lots of money and no students. So I went out and talked to Jim, and we hit it off pretty well. And I think he sort of viewed me as sort of a pity case because he let me join his lab, even though I had never worked in a lab in my life. And he took me into the lab, and this was in 1983, in the fall of 1983. And I'll just point out a little correction from yesterday. Lauren was actually Jim's first master's student. I was actually Jim's second PhD student. Linda Atkinson had joined the lab about 6 months before me. And so we were sort of the first two PhD students that had come through Jim's lab. And shortly after I joined Jim's lab, I was watching TV one Sunday night. They had this network premiere of Star Wars. Do y'all remember when they used to advertise the network premiere of sort of the greatest movie back when we had free TV? And Star Wars was on. And so I was watching it, and they always had these news breaks during Star Wars. And I was just fascinated. I was, a science geek, so I was watching Star Wars, and this news break came on, and it really caught my attention. And so I actually, just be careful. Anything you do in your life, you can find on the internet nowadays. And so we're going to play this clip that I actually found on YouTube. Hopefully it'll play here.
Good evening. From CBS News, this is Newsbreak. After 17 months, a scientist at Texas A&M says humans and cows are a perfect match in gene traits. And the cows can be used instead of mice in experimental research. It may seem like a good idea in Texas. Elsewhere, mice may seem easier to get into those little cages.
I never said it was Jim Womack. When I was watching this, I knew immediately who they were actually talking about. And that was in February 26th of 1984 that actually came on. It took me a couple of years to actually find that clip online because I couldn't remember when it was or anything like that. But when I joined Jim's lab, the lab was still doing a lot of starch gel electrophoresis, a lot of isozyme work. And so I learned how to do a little bit of that. But Jim basically said that Linda Atkinson and I need to develop molecular tools because that's where he wanted to go. And so in 1984, he actually got a new lab space in the basement of VMA. So those are familiar with the vet complex. And in the basement of VMA, he locked Linda and I in the room and said, okay, I want you to develop southern blots and sort of to be able to do molecular stuff for the mapping. And after about 2 years, we had no internet, trying to figure out how we actually do this. We finally got our southern blots to start working, and Jim came down there, unlocked the door, and let us out. And we got to move up to the big lab then, and that's really when I think the lab sort of exploded. Because by that time he had hired a bunch more students and postdocs. We had a very large and active lab at that time. So my research ended up being on the bovine major milk protein genes. And I really focused on sort of using these new molecular tools that I had to develop in Jim's lab to really address the structure of the milk protein genes in the genome. And this required, developing the southern blots for SSLP analysis so we could look at polymorphisms across the different cattle breeds. I developed a partial digestion 2D pulse field electrophoresis. This is before we had the ability to do the radiation hybrid, so I was using pulse field electrophoresis and partial digestions to actually look at long-range maps of what the order of these genes were actually in the genome. And we actually developed the first in situ hybridization on cattle chromosomes before Rudy Fries had developed the really nice fluorescent ones. We were using I-131. And I probably labeled my thyroid at least a couple of times during these experiments. But suffice it to say, we got all of these techniques to work. And I remember Dan Gallagher and I sitting at a table cutting out cow chromosomes at one time, trying to figure out how do we identify one of these 60 acrocentric chromosomes. And it's not like these chromosomes were big spreads. You know, they were little tiny chromosomes. And it was a chore trying to identify these with these little black spots from the I-131 that had labeled the chromosomes. But, you know, I have to say it was a really, really good time. It was probably the best time in my career being in Jim's lab. I got to meet many genomic pioneers that came through the lab. Many of them are here. Steve O'Brien had come through. I remember one time I was in the lab really, really late at night, and Jim and Steve were in there trying to polish up an annual reviews document. And I think they must have sat in the lab all night doing that. I met Rudy Fries, Michelle Georges, Leif Anderson. All of these characters had come through the lab at one point or another. And so it was just a really great time during that. About 2 years in, Jim and I actually made a very unusual connection. It actually turned out that my stepdad was Raby Womack's high school basketball coach and math teacher. And you know, after that, I think Raby sort of took me under her wing and wanted to make sure I had an appropriate life after science. And I remember one time we had a barbecue over at Jim and Raby's house, and this was the first time Debbie and I had shown up together. We'd been dating a couple of months, so Debbie was also in Jim's lab. And little did I know that Raby had invited her student teacher that she was working with to the party to introduce her to me. So needless to say, it was a little awkward at that party because here you had the student teacher that Raby was trying to introduce to me, and I had shown up with Debbie, and so it was an interesting situation. And I'll also say that, you know, in my time in Jim's lab, I actually was still doing a lot of the mouse genome work. In fact, my first 3 papers in Jim's lab were actually mouse mapping experiments. And so I was still sort of doing the mouse work as well as doing some of the cattle work. And most of you may not know why Jim actually got out of the mouse work. And Provost Sams, this is a good reason why we need a new vivarium. We still were using LAR, the Laboratory Animal Research Building at the time. It was a relatively new building. And we didn't have good HVAC control systems. So over the weekend, the HVAC went out and the rooms shot up to about 90 degrees. And all of Jim's mice were roasted. And we came in one morning on Monday, and basically the mice colony had been wiped out. And that's, I think, pretty much what drove him out of sort of this dual mouse cattle world and really had him refocus just on cattle at that point. But I have to say that, you know, Jim was the type that you never saw him get down. You know, you never saw him get upset. He was such a even-keeled person. Even in that event, at least he didn't show it to anybody in his lab that, you know, he was really bothered by that. And I've really tried to model that in my career because, you know, I don't think you could have asked for a better mentor than what Jim had. Another story that I'd like to say, so a couple of times Jim and I went bass fishing at my family's ranch, and Jim was a big fisherman. You know, he really liked fly fishing. And he and Lauren actually had a trip scheduled out to Utah to go to a biochemical genetics workshop. And right before that is when Jim had his heart surgery, and he had to go in for quadruple bypass surgery. And I think Lauren was a little disappointed because Jim asked me if I would stand in and actually go fishing with Lauren. So we went to Flaming Gorge in Utah to go fly fishing. I'd never fly fished in my life. So here, spending 4 or 5 days fishing with Lauren, here I am trying to use a bass fisherman's approach to set hooks, which doesn't work when you're fly fishing. I think Lauren was just wondering, when is this going to get over? But I have to admit, it was a really fun trip. I got to learn a lot more about Lauren. But Debbie and I had actually gone down to Houston to see Jim in the hospital right after his surgery. And, you know, I think this is really indicative of someone that's just a scientist at heart. He was not concerned about his health. He wasn't concerned about anything like that. He was just fascinated by the technology of vascular replacement surgery. And so we were sitting there talking with him, and he opens up his shirt to show us his stitches on his chest, because he was so proud that they had opened up his chest and done all of this work. And he was just, excited about the technology of that, much more so than what he thought about, what the impacts of his health would actually be. But speaking of his heart surgery, I'm not so sure that it wasn't the graduate students in the lab that actually contributed to that. Because it turned out, I'd mentioned that we'd actually gotten a very large lab at that time. We probably had 20 people in the lab. And let's just say there were quite a lot of practical jokers in the lab. And I'm just going to say one thing, and that is I think Donna Musnu was probably the instigator to most of these. She was the ringleader. And so if you want to hear any interesting practical joke stories, you go ask Donna about this. She's probably got a long list of these. But there was a lot of us actually involved with this. And it was just a really, really fun time in his lab. So, getting to Bill now. And so the reason I sort of wanted to give this introduction about Jim is that many of the traits that Jim manifests, I actually see in Bill. And I think Jim is a good example that good guys do finish first. This is someone that just had a good heart and that was just really a tremendous individual. And so I was familiar with Bill's work from the time he was actually in Steve's lab. And I hadn't met Bill, I don't think, at the time, and we may have crossed paths somewhere, but I was familiar with a lot of the work he was doing in Steve's lab. And currently, and I really didn't get to meet Bill until about 2013 when I moved back to A&M. And we've gotten to be really good friends and colleagues and sort of integrated into the genetics environment. So Bill currently holds the James E. Womack University Professorship, which I think we couldn't have asked for a better person to hold a professorship in Jim's name. He really is the ideal person to sort of take the mantle of comparative genomics and really take it to the next generation. And his new center on comparative genomics, I think, is really going to revitalize the activities in comparative genomics at A&M, is really going to have a large impact. So Bill has really been sort of a new generation pioneer in comparative genomics, really looking at it much more broadly just because of the tools that are available than we really had in the past. And all of Bill's accomplishments, he's really sort of, I think, starting to come into light and really be appreciated for everything he's done over the last year. He's had quite a year in awards over the last year. So he was named a University Distinguished Professor, a Texas A&M Assistant Regent Professor, and he was elected to the National Academy of Sciences all in the last year. And I think these are well-deserved recognitions that Bill was way overdue in receiving because of all of the contributions he's made. And really, Bill does exemplify many of the traits that Harris Lewin talked about that Jim has. Besides being an outstanding scientist, he's very generous with his time, his reagents. He's an outstanding mentor to his students, and he's just really a good guy. So, someone that's usually a really good person, there has to be things in the closet. So I actually had asked Steve O'Brien to send me some really juicy stories about Bill so that I could sort of use some of these stories. And the problem is, Steve sent me a list that I don't think we have time to go through some of these embarrassing stories, Bill. But I'll just stop there and let Bill tell us about his career and his interactions with Jim. And it's just really a pleasure to have Bill talk today.
Thanks, David. That was really kind of you. It's an extraordinary privilege to be here leading off this morning's talks. And as he said, it's been a huge privilege for me to have worked with Jim and many of the colleagues here over the last 20 years. He's been extremely supportive of me. When I was told that I could have a named professorship, the obvious choice was Jim, because it had to be someone who was retired and in your discipline. And it took me about 2 weeks before I summoned up the gumption to ask him. And he, of course, was extremely generous. So that was just one of many things he's done for me throughout my career. And I'm extremely grateful for being able to work with him. And I will actually highlight some of those many ways in which he has advanced my career over the last 25 years. So Lauren asked me to talk about the dark genome. And many of you might know what that is. It's sort of a niche sort of term. It's been thrown around a lot, and it sort of relates back to many different facets about the parts of the genome that we don't understand. And I think it started obviously with the human genome, and when we first sequenced the human genome, well, there's 2% that's protein coding genes, and what is the rest doing? And that's really where the value of comparative genomics has come into play. And comparative genomics, obviously, I think Harris introduced the field very well, and many of you know very well what comparative genomics is, but for those of you who may be new to the discipline, you can sort of capture it through a number of different lenses. And I'm going to steal this slide from NHGRI that I think is one of my favorites, that really sort of outlines the many different approaches and lenses through which comparative genomics can be tackled. And so the traditional approach that I think many of us are accustomed to is the idea of basically comparing very distantly related organisms to identify key evolutionary conserved elements. And I'm going to touch on that component early on in my talk in terms of what are the best approaches to do that. What did we learn from using comparative genomics and phylogenetics to help solve some of those questions? And then I'm going to go to the next level and look at through the aspect of looking at closely related species. And there's been a lot of really good work recently on comparing great ape genomes to the human genome, highlighting some of those aspects. I'm going to talk about cats because, thanks to Steve, that's what I spend most of my time doing. And then, of course, actually nowadays, population genomics is considered comparative genomics, this genotype to phenotype mapping. But genotype to phenotype mapping can be discerned through all of these different levels of comparative genomics. And I'm going to spend my time at the deeper level, jump down to closely related species, and then go back to the deeper level based on a newer type of comparative mapping that we've just been recently doing in the lab that has been producing some exciting insights. And so I was extremely fortunate to be at the right place at the right time. And maybe Steve had pity on me. He knew I came from a PhD lab where I was working on fish phylogenetics to better understand chromosomal evolution. And of course, he said, Murph, you're not going to work on fish in my lab. You know, we work on mammals, and you're going to work on cats. And so my first assignment that Steve gave me was to build the first ordered genetic map of the cat genome. And the best way to do that at the time was using radiation hybrid mapping. And fortunately, Steve was good friends with Jim. And as Harris pointed out last night, Steve connected with Jim, and Jim graciously sent his protocols to myself and Mary Thompson, who together we produced the first radiation hybrid panel. And that was one of the several panels that I actually developed in Steve's lab. We also went on to develop a macaque radiation hybrid panel and some other interesting mammalian panels, including an elephant that I won't talk about. But basically that passion for using radiation hybrids to look at genome organization was something that was really exciting, and because of Jim's generosity really started my career and really galvanized my interest in comparative genomics and really understanding the functional coherence of chromosome organization. And I will point out, I'm not going to talk about this today, but actually after I joined Texas A&M, Jim's lab and my lab actually teamed up to create 5 radiation hybrid panels from a number of different species for high resolution mapping for Y chromosomes in a collaboration with David Page's group and many of his collaborators. So that was another exciting time where I got to work closely with Elaine Owens and Jim's lab to work on our passion for comparative mapping. So this is the paper that actually Harris showed last night that really got me excited. So I had created the first radiation hybrid panel for cats, and this paper came out in 1998. I had finished the panel about a year before, and I saw this paper and I was like, that's what I want to do. I was extremely excited, the idea, and of course their example chromosome showed a number of inversions and different rearrangements. And so the first chromosome I picked was the X chromosome. So there was some interest in sex chromosomes at LGD at the time, and I guess in some ways a little bit of a disappointment. The X chromosome was a little boring in terms of, we found that the domestic cat X chromosome was extremely conserved. In fact, of all the markers that we were able to map, they showed identical gene order between human and cat. And that was a real exception because at the time, all of our comparisons were really ordered comparisons were between mouse and human. And I'm not going to show my slide to not insult David today, but why mouse is not a good model. But one of the reasons is the mouse genome is exceptionally rearranged. And what we found over our careers is that basically the mouse in many cases is an exception. Obviously it's an excellent model, but there are many cases in terms of chromosomal evolution and other topics I'll discuss today where it's kind of an oddball. So this is really what really stuck with me for a long time, because this observation that an entire chromosome could be absolutely conserved over 95 million years was actually pretty remarkable at the time, right? There was no other autosome where we observed this type of conservation. And my colleague Terry Raudsep worked with Jim and Banu Chaudhary and Lauren to build the first equine radiation hybrid panel. And they, shortly after this paper, showed the same thing in horse. We actually had some unpublished data from macaques and elephants at the time that showed they were also conserved. So in my mind, I always wanted to know what was driving this conservation of gene order. The conservation of gene content on the X chromosome was expected. Susumu Ono predicted that the X chromosome would be conserved because of functional constraints due to dosage compensation. Females have X inactivation, and because of that, translocations and movements between autosomes in the X chromosome would be deleterious. And so, but that of course did not explain why the gene order itself should be conserved. And so I'm going to come back to that as sort of this long-standing quest in my lab to really understand the nature behind why this particular chromosome was so remarkably conserved. But again, I want to point out that being in Steve's lab, I was introduced to Jim, I was introduced to Jenny, I was introduced to many of you, especially Harris. And I think I was trying to think about this last night, Harris, after your talk. You know, when did we sort this out? And I think we met at a PAG meeting, probably around 2003. We had seen each other's papers, and we decided that we were going to sort of join forces. And obviously, with Harris working with Jim, Steve knowing many of the people in the comparative mapping community, we got together this group. And in fact, in 2003 or January of 2004, we had a workshop in Pavel Pevsner's lab at UC Santa Cruz, where the whole group who was at the PAG meeting, the pig group, the Larry Shook here, the dog group, the cat group, the horse group, and the cow group, and we sort of set out a game plan as to how to do some comparative mapping, taking advantage of a few genomes that were finished, the mouse, the rat, and the human, but then using the emerging comparative maps from these other species. And so this was just an exciting moment to be able to collaborate. And I actually got to really know the Texas A&M group quite well. And during this time, I was recruited to A&M, and in no small part because of Jim's influence that I decided to come here, and certainly don't regret that move. But a few years after the human genome was completed, obviously the human genome was announced with great fanfare. And as I pointed out before, the big question was, what was the rest of the 98% doing beyond protein coding genes? And which parts matter, and it can't be all junk. And junk might be, you know, the old term for what we call dark matter nowadays, but I think dark matter has sort of evolved in terms of its terminology, and I'll come back to that. So these questions were related to, you know, how would we choose beyond mouse and rat the next best species? And as Harris pointed out, there was a lot of controversy about which of those would come next. Would it be the dog, which was first, then cattle? Cat, unfortunately, came later. What were the motivations? And I wanted to correct a little bit of the history that Harris gave last night. He kind of poo-pooed this CATS primer idea. And I actually am a fan of the CATS primers, right? There's not many things I'm good at, but one thing I'm pretty damn good at is designing some conserved comparative primers. And because I was really good at that, we were able to use this approach, using the ones that have been designed in Steve's lab in collaboration with Jim and Neil Copeland and Nancy Jenkins, but also design a bunch more. And we use those not just for building gene maps, but because at the time, Science Magazine had this genome issue on comparative genomics. For the first time, comparative genomics was really in the spotlight. And Steve tried to get together a group of paleontologists and molecular biologists to agree as part of this poster on the phylogeny of mammals, because the phylogenetic history would largely guide some of those next choices. Well, long story short, half of the team quit. The paleontologists couldn't agree with the molecular biologists. The molecular biologists couldn't agree. And this was a big unsolved question. And so we, myself and Eduardo Eiserick and a couple others in the lab, teamed up with Ollie Ryder's help at the San Diego Zoo to basically try and provide this phylogenetic framework for comparative genomics. And so we published in 2001, the same year the human genome was released, this phylogeny. And one of the important things that it revealed was that human and mouse are just one of, hold on a second here, are just restricted to one of 4 major lineages of placental mammals. And we argued in this paper at the end that expanded genomic sampling beyond that clade would actually be extremely important for understanding the human genome. And actually, not within a week or two after that, Eric Green came up to the lab and basically said, hey, how are we going to use this information? And then Steve said, let's write an opinion piece. And we sent this into Science, sort of discussing, based on this phylogeny, what would be the motivations for selecting species for comparative genomics. And of course, that included biomedical relevance, economic importance. Were they phylogenetically close to human, like chimp, or were they phylogenetically distant? And so basically that motivation to sequence more mammals to help annotate and understand the human genome is really one of the reasons that the drop in sequencing costs came down, invigorated new technologies, and ultimately led to an explosion in genome sequencing over the last few decades. And perhaps in no other part of the tree of life has this accelerated than in mammals. And so I was very fortunate, having been involved in comparative genomics in mammals and having worked in phylogenetics, to be invited to be part of this Zoonomia consortium that I'll talk about here in a second. And so mammals are obviously an attractive target because they have this incredible diversity. They unrivaled amongst any vertebrate clade and many other large clades of animals. And because they all share roughly 19 to 20,000 protein coding genes, with obviously some tweaks of gene families, gene gains, gene losses, mutations, that's led to this great variety of form and function and ecology. And so when we're trying to understand what are the commonalities amongst these weird forms, comparative genomics can be extremely powerful, but also, if you're interested in any trait, just go out and look at the over 6,000 species of mammals, pick the one that matches your trait of interest and see if you can use comparative genomics to solve that. I think we're actually finally in a position to do those types of studies. So I was excited to be able to use the resources of the Zoonomia project, which was a 240 species genome alignment. And the motivations, of course, were human-centric. It was to guide the interpretation and annotation of the human genome. And Shostin Lindblad led this effort many years ago. The first was the 29 Mammals Project, where they sampled deeply across the phylogenetic tree to help annotate the human genome down to not base pair resolution, but close to that. But the goal of the Zoonomia project was to get to base pair resolution and to discern from that alignment which bases were under strong evolutionary constraint, which ones were neutrally evolving, which was valuable for our phylogenetic purposes, and then those that were accelerated. And so obviously to do this, we had to first estimate a phylogeny, but we also took advantage of this new approach for genome alignment. So in the past, most comparative genomic approaches would always align to a single reference, and that of course introduces reference bias. If you're aligning to the human, you're not going to find any sequence in those other genomes that happens to be lost in humans, right? So this progressive cactus alignment basically allows you to see different layers of shared ancestry across the phylogeny, but the beauty of the Zoonomia alignment is that you can project this alignment to any reference you want. It can be to the human genome, it can be to the dog genome, to the cat genome, and we're doing this right now to project this to the cat genome to better understand what are the parts of the cat genome that are novel and species specific. Are there parts that are conserved that were not with other mammals that were lost in humans? And so this is an extremely powerful resource that we're only beginning to explore. But to do this, we needed a phylogeny, and along came the wonderkin, where is she? Nicole Foley, over there, Dr. Foley, who came from a former colleague of mine, Emma Thieling's lab, and she was very interested in phylogenetics, and so she was lucky enough to tackle this whole genome phylogeny. And so this basically was a framework. And the reason why I showed this particular slide is it's this sometimes underappreciated concept is that the rate of evolution across mammals is extraordinary, right? So you go back to the ancestor and you measure the length of these branches, that's the substitution rate. And it's highly variable, highly predictable based on generation time, body size, metabolic rate, right? And so we've got shrews, and rodents and mice are the furthest out there. They are twice the rate, their mutation rate is twice the rate of most other placental mammals. But this density of phylogenetic history and mutations is really what comes into play when you're trying to understand and annotate the human genome. Because with this dense of sampling, we have basically allowed that every site in a conserved alignment with the human genome will have had an opportunity to have mutated many, many times throughout the history of mammals. And so if we search in the alignment, we're going to obviously find that there are many sites that are conserved. Even though there was an opportunity, those sites that mutated in basically critical regions, critical for development or other reproductive functions, those would have been removed due to purifying selection. And so those signatures are found in conserved alignments where we have these mammalian conservation scores shown across the human genome browser. Here's a typical gene shown here. You expect to see conservation over protein coding portions of the genome, but you don't necessarily expect to find very high peaks outside unless they're critical for regulatory function. And so this approach, which again was started by the 29 Mammals Project and then expanded upon with the Zoonomia Project, has been extremely powerful and is probably one of the best ways to infer function. And so what it did was took from what we thought was about 6% or 5% constraint outside of protein coding genes to almost 11 to 12% constraint. And so that was a real novel finding, provides a lot more candidates for potential functions when it comes to regulation. But how is this really useful? I'm gonna pitch to Scott Dindo here in the audience. I'm not going to give a talk on Angelman syndrome, but Scott actually, I think, was one of the first translational examples of using comparative genomics. And I'll point out that Jim was on his search committee, and I think Jim would be extremely proud to know that Scott, using comparative genomics, was able to define a highly conserved region that hadn't really been identified before, that he could design an ASO for that would be a therapeutic for Angelman syndrome. And it turns out that happens to be in a phase 3 clinical trial now. And so I think Jim is smiling now to think that probably the best translational application of comparative genomics came from Texas A&M University. So again, it's an exciting example and a testament to how those types of approaches of looking for functional constraint actually have real life applications. So to the human T2T genome, this was released only a few years ago. Getting back to the dark matter, one of the really exciting things about the human genome, and I stole this from NHGRI as well, is that it took forever to finally complete the human genome. Even though it was announced in a draft form in 2001, it wasn't until about 2022 that they actually completed that last 8%. And what was added? Well, it was all of this dark matter that hadn't really been assayed before. We knew it was there, but centromeres and ribosomal RNA arrays, large tandem arrays, segmental duplications, things that we knew about but couldn't accurately genotype because they were either never fully assembled or they were only partially assembled. And so this dark matter was new to the human genome. And of course, we have to assume that in any other genome that's not truly T2T, we're missing all of these sequences and probably more. And so I think that's critical for comparative genomics, because if we're going to compare apples to apples and not apples to oranges, we have to have a complete picture and be comparing the same quality of references to one another so that we're not making false inferences about this species has more than the other or not. And so how do we get there for other species? And so this goes back to cattle and the cattle genome. So it was Sergei Korin and colleagues that developed this idea of using trio binning in F1 hybrids. So in comparative genomics, many of you might remember that F1 hybrids were key, or at least thought to be a valuable component early on because of the increased polymorphism between subspecies or species. It would make mapping genes or protein coding genes much simpler. And so turns out that many years later, this trio binning approach applied to F1 hybrids was incredibly powerful at resolving sequencing gaps that were found in diploid-based pseudohaplotype genome assemblies. And you can see this yak and cattle genome, each of these single gray or black bars indicates a complete contig representing a chromosome. So this was exciting. I remember seeing this at a PAG meeting, and we were sitting around, myself and Leslie Lyons and Wes Warren, and we're thinking, oh man, we actually have a lot of hybrids in cats. This would be extremely powerful. And so thankfully, Steve's lab had been looking at interspecies hybrids for quite a long time. In fact, cat hybrids have been quite instrumental to many different parts of the cat genome story. And so the Bengal cat, for example, shown here, which is a hybrid between the domestic cat and an Asian leopard cat, diverged about 6 million years ago, were used to create the first genetic maps. And then this interspecies cross between a domestic cat and a Joffrey's cat diverged about anywhere from 8 to 10 million years ago. They were actually used in the 1980s and 90s to study hematopoietic stem cell research. Ligers, of course, we didn't have those at LGD or at Texas A&M, but they are bred some places and provide another opportunity to develop genomes for endangered species. So the other exciting thing about studying hybrids, and as an evolutionary biologist by training, is this idea that most mammals, and cats in particular, follow the 2 rules of speciation. And there aren't many rules in biology, and one of those is Haldane's rule. And this is when in the F1 offspring of 2 different animal races, if one sex is absent, rare, or sterile, it is always the heterozygous, or what you know as the heterogametic sex. So in mammals, that's the male, the XY. And the second rule that is related to this is the large X effect. And that we know that hybrid incompatibilities tend to accrue on the X chromosome due to evolutionary divergence between the parent species because of the hemizygosity of the X chromosome, and that these alleles are exposed. And that's why males are affected disproportionately relative to females. So this is an exciting opportunity to use genetics to explore the genetic basis of speciation and reproductive isolation using hybrids. And most of the work that had been done in this field, actually for many years, had been done in Drosophila. And actually, surprisingly to date, only a handful of genes have ever been identified that are thought to be true speciation genes, if you want to use that term. And only one in mammals, in mouse, the PRDM9 locus. So cats exhibit these typical traits that we see in other mammal interspecific hybrids. They have this meiotic or pachytene arrest, where basically we see cattle hybrids showing the same manifestation, rodent hybrids, the same manifestation. And studies in rodent models had shown that this particular phenotype is due to disruption of this XY body, which is a heterochromatized structure where the X and Y chromosome are largely silenced during the pachytene stage. And it's thought that the disruption of this sex body is what ultimately results in this pachytene arrest. And so this is the current hypothesis for rodents. And it turns out we had evidence in cats across multiple interspecific hybrids, work from Brian Davis and Wes Brashear and Kevin Bredemeyer in my lab, showing that across all the hybrids, indeed, we see a upregulation of the X chromosome in sterile versus fertile hybrids. So when we're comparing gene expression, we see, again, this consistent biomarker of the X being upregulated, which is likely the manifestation of the loss of the XY or the sex body, right? That's the hypothesis. And why is this relevant? Well, back to trying to understand the genetic basis for speciation, what we know from Drosophila work is that many of the genes that had been identified evolved really rapidly, are highly polymorphic, and are often in satellite elements, parts of the genome that are known as dark matter. And why is that relevant? I'll get to that shortly. But we wanted to use this trio binding approach to resolve the dark matter of the cat genomes and sort of get at some of these questions. And so here's an example of how we use this approach. We have an F1 savanna hybrid. These 2 parent species, the domestic cat and the serval, diverged almost 10 million years ago. It's one of the most divergent mammalian interspecific hybrids. Yet they produce, without AI, hybrids in captivity when the domestic cat isn't eaten by the serval. But the beauty of this approach is that if you have an F1 hybrid where the parent haplotypes are extremely divergent, you sequence the F1 with long reads, you sequence the parents with short reads, and basically those k-mers essentially serve as barcodes to phase those long reads before genome assembly, so that the genome assembly process is not confounded by this problem with heterozygosity that often causes assemblies to break or produce completely erroneous results. So here's just a few examples of this F1 approach. Basically, clean down the middle, the newest hi-fi sequencing, we're able to resolve all but a single read phased between the two parent genomes of this F1 savanna. So it's a highly accurate approach, and because of that, it yields highly complete near T to T genome assemblies. We're very close to T to T in many of these cats. I'm not going to talk too much about those today, but Andrew Harris here has been working on that. So this is an exciting time for us in the lab to be able to start to explore some neat parts of the genome. So what were we missing? We kind of know from the Human Genome Project, in cats we're missing the same sort of things, centromeres and large tandem arrays and complex gene families that are relevant to traits related to reproduction, immunity, sensory biology. So I'm going to focus on one of those today, back to hybrids. So to better understand the genetic basis for hybrid sterility and species formation, we were actually trying to use a particular hybrid breed called the Chowsy, which is a cross between a domestic cat and a jungle cat. So these species diverged about 3 million years ago. They follow Haldane's rule, and the breeders are extremely generous because they have animals which they've kept or have good breeding records for which we're able to breed, or which ones were not. They were able to provide us testicular samples to validate that many of these males that they thought were infertile were indeed showing that Pachytene arrest. And using a genome-wide association approach, we were able to map a single major effect locus to the X chromosome. And we were able, through fine mapping using some polymorphic sign insertions between those, to a fairly narrow interval on the X chromosome, that lo and behold, in the assembly we had an assembly gap. And we sat for years trying to assemble this with the newest approaches, even using back clones, and boy, some of my graduate students just hated it because, you know, even with even with Oxford Nanopore, it was very noisy. We just could not get through and assemble this region. But when we finally were able to, with these trio bin assemblies, we were able to reveal this full structure. What ended up being the most compelling candidate, these two protein coding genes on either side were not compelling for a number of functional reasons. Either weren't expressed in testes or had no known role in spermatogenesis. But DXC4, this satellite array, was quite intriguing. Turns out it was only finished for the first time in the human T to T assembly. So again, these are parts of the genome that we know are highly polymorphic. They're rapidly evolving, but they're almost rarely explored or genotyped because of this structural complexity. And we were able to infer that between the parents, using some indirect estimates, that the domestic cats had a lower number of copies than we could guess from, that we could infer from short read mapping compared to the jungle cat. So the two parents did conceivably have a copy number difference. Well, why is that relevant? Well, we know quite a bit about macrosatellites from studies in human. Perhaps one of the best studied human syndromes due to a macrosatellite-based disorder is facioscapulohumeral muscular dystrophy. It's due to copy number variation at the D4Z4 satellite array. And what happens is that in those patients, different length arrays basically induce a copy number methylation-based effect that can result in the misexpression of genes in the wrong tissues at the wrong time, basically. So macrosatellites are sort of attractive because, in terms of a speciation locus, because several of these have been identified in Drosophila as the genetic basis for some of the early hybrid sterility genes in Drosophila. So they have highly variable copy number. They're enriched for CPG islands. They have this copy number threshold that's been described for many affecting methylation. And there was some literature about DXC4, and it related to X chromosome biology, obviously. And it had been observed during X chromosome inactivation that DXC4, like exist, the transcript that's expressed and silences the X chromosome, is silenced on the active X chromosome. But on the inactive X chromosome, it escapes silencing And DXC4 is interesting in that if you looked at high C data, a typical active X chromosome looks like any autosome with these TAD structures, but the inactive X chromosome showed this remarkable bipartite structure, this sort of lack of TADs, but these two huge domains. And this DXC4 hinge-like structure was conserved across mouse and human and other primates. We showed in cats it was the same, it was also conserved. So it was an intriguing candidate. What was interesting about DXC4 is it interacted in 3D space with other satellites spread across the X chromosome. ICE, the X inactivation center, and another one called FIRE. Now, because it was expressed during X inactivation, it was thought that if you disrupted DXC4, it actually would disrupt potentially X inactivation. Sadly, that isn't the case. So we don't really fully understand what DXC4 and some of these satellites are doing. But again, we do know that they are involved to some degree in the 3D organizational structure and possibly associated with removing the bar body away from the active X and towards the periphery of the nuclear membrane. So you might be asking, well, why is it a good male hybrid sterility candidate, right? We're talking about X chromosome inactivation. Well, as I told you earlier, there's some striking parallels between meiotic sex chromosome inactivation and X chromosome inactivation. Parallels, not exact similar marks or mechanisms, but the XY body also forms this heterochromatomized structure that is isolated and removed from the autosomes during meiosis. And the same regions of the inactive X during spermatogenesis and female somatic cells are shown to have the same escapee marks in these two different chromosomes in these two different circumstances. So we thought it was actually an intriguing candidate, but no one had ever really discussed or explored what DXC4 might be doing during male meiotic sex chromosome inactivation. So my graduate student Kevin basically was able to work with John McCary at UT San Antonio, to get purified germ cells from domestic cats. And we were able to show that indeed DXC4 was expressed at exactly the time when the X is supposed to be silenced. So it was escaping X inactivation at the time when we see the sterility defect. And he further showed that in sterile hybrids that DXC4 was downregulated, and that in sterile hybrids, the first repeat array, DXC4 repeat unit A, was hypermethylated. And we speculated that this was due to the copy number difference between the hybrids carrying the jungle cat allele and the domestic cat allele. Most sterile hybrids carry a jungle cat version of DXC4, and all fertile hybrids carry a domestic cat version. So taken together, that makes sense that likely a copy number mediated effect might be driving hybrid sterility. But also, we took that a step further and compared the rate of evolution of DXC4 to the rest of the genome. And what we found is that it's actually evolving as fast as 95% of the rest of the genome. So these repeat arrays are some of the fastest evolving parts of the cat genome, probably of mammalian genomes in general, and they make sense as a speciation gene in that you'd expect speciation loci to evolve on rapidly evolving genomic templates. But what's also intriguing is that this repeat array and many of those other satellites are conserved across mammals. So we were able to dig through, again, incomplete assemblies and show that there's actually differing array types. So cat had two repeat arrays. We had an A type and a B type. Humans only had the A type, but other species, based on incomplete assemblies, we could garner had A or B. So it's conserved, but it's not only conserved, it's conserved in size and CTCF sequence, but not the rest of the repeat array. So that's an interesting part of comparative genomics that's not really tackled or discussed, is why is it that you would have this functional satellite that keeps the same repeat unit size despite complete turnover in its genetic sequence, except at the CTCF binding site, right? That's something we wouldn't be able to explore without comparative mapping or comparative approaches. But also intriguingly, these sites look to co-evolve. So if you look at DXC4, the copy number between DXC4B, DXC4 is known to interact with ICE, and it also has a copy number variable repeat number, and it seems to co-evolve in that longer repeats exist in ICE and DXC4, and they shorten across species lines. So there does seem to be some potential co-evolution of these satellites, and it's perhaps incompatibilities between those loci that are driving speciation. But getting back to my early question about why is the X chromosome conserved, well, these loci have to interact in 3D space. And so we suggested that perhaps selection against chromosome rearrangements, altering these 3D interactions, actually might actually disrupt X chromosome inactivation or whatever these satellites are doing. And so we don't know what's driving this constraint, but what's really important is that if you look at mouse, which differs from the human genome and these other conserved genomes by almost 10 inversions, basically the order and the spacing of these satellites is almost the same, right? So again, it suggests that these could be part of the reason why the X chromosome is highly conserved in gene order, because they're maintaining these cis-acting interactions. And now, thanks to comparative genomics, Nicole has been able to show that across placental mammals, this conservation of X chromosome gene order is largely conserved, right? And so again, is this satellite interactions driving that? Possibly. But one other thing we observed in the lab studying cat speciation and looking at how genes flowed between species boundaries was that we had been building recombination maps with Steve's help using a SNP-based linkage map. And we showed on the X chromosome that basically the X chromosome recombination landscape, this is the recombination map on the y-axis and the genome map on the x-axis, that you can see that it has this striking undulating recombination profile, where you have low spots separated by hot spots and cold spots and hot spots. But it was remarkably conserved. We went digging through the literature, and sure enough, pig and human independently, these are their figures, highlighted these same regions, right? So that's remarkable, because recombination rate in the literature is thought to evolve very rapidly across evolutionary time. In some lineages, it's a bit slower, but we don't expect this undulating intervals of high and low necessarily. It's one thing, for example, in bird genomes, we see many large chromosomes tend to have very low recombination rates at the center and high at the ends, but the X was remarkable in this highly variable landscape. So we took upon a new project on comparative recombination mapping. And so a new way to sort of look at this other dark part of the genome that hadn't really been explored across a deep time. And so Nicole and her students mapped 48 billion reads to 22 long read assemblies. We used this new machine learning approach to build recombination maps from population data. Obviously, we weren't going to get pedigrees and be able to assay recombination rates across divergent lineages like elephants and whales, but with these new tools and recombination based mapping with these machine learning approaches, we're able to produce remarkably precise recombination maps that they match what we see with typical pedigree-based linkage maps as well. But what I want to point out, again, relevant to why is the X chromosome conserved, is that in lineages of the conserved collinear X chromosome, we see that this central cold spot spanning almost 50 million base pairs is absolutely conserved and is low across most mammals. And if we distill the chromosome down into quintiles of really cold and really hot spots, we can see that this cold spot density is conserved across placental mammals, and the hot spots are even conserved. So again, why is this? This is sort of remarkable, something we've never really seen. And then taking it another level, Nicole, basically for those same lineages where we had recombination maps, she went out and built phylogenetic trees for groups of species from each of those lineages, and look to see where in the genome was gene exchange or interspecies gene flow permissible, and where was it not? And what she found is that, as you'd expect, in lineages that we knew had evidence of gene flow, that when you found ancestry trees across the genome that were introgressed, they were typically common on the autosomes and depleted on the X, and especially depleted in the middle of the X chromosome. And if you look again across placental orders, this XLRD or this conserved low recombination desert, as we call it, is highly conserved. It is a consistent barrier to gene flow. and we've looked at many different mammal lineages, and we've yet to see cases where there's a lot of genetic exchange happening in this region. So is it a supergene? Leif Anderson might have thoughts about this. He's done a lot of work on supergenes. Sex chromosomes by their nature are considered supergenes. They're widely interpreted as consisting of multiple tightly linked loci that are controlling or regulating a system of discrete phenotypes. And again, we don't really know exactly what might be the components of driving this supergene, if it is a supergene, but there's some intriguing candidates. So as I said before, we have the potential cis interactions between those satellite elements. But also, if you look at enrichment for different categories of genes, there are several classes of loci with sex-biased or sex-restricted functions that are enriched and spatially dispersed within the XLRD. So we have an increased density of testis-biased expression and ovary-biased expression within the XLRD. We have basically the cis interactions between macrosatellites that are expressed in female exon activation and male meiotic sex activation. And then intriguingly, testosterone loci in males are actually enriched in the XLRD. So sex-specific GWAS performed on human males and females actually produce extremely different genetic architectures. It's actually like they're completely different traits. And so there's very little overlap between loci controlling testosterone levels in males and females, and it turns out there's a strong sex-specific effect. Loci enriched on the X chromosome, well, not enriched on the X chromosome, but at least enriched within the XLRD, there are no X-linked loci in females that are associated with testosterone levels. So again, we're not suggesting that any one of these is responsible for these effects, but there's certainly a plethora of candidates that could be driving sex-specific effects and sexually antagonistic effects that might explain the suppression of recombination. So in conclusion, be mindful of the dark side. There's a lot of very interesting features that we're able to now ascertain from genome assemblies. And this complex repetitive variation underpins important phenotypic variation that we're only now beginning to explore. And I think a really unappreciated and untouched area, these sort of sequence-based measures of conservation, like spacing and even recombination rate evolution that we don't really understand what's driving their conservation. So I think there's a lot of very interesting biology to be learned over the coming years with the ability to sequence genomes to completeness and across the tree of life. But my final conclusion, of course, is what Jim said, is mapping genes is good for you. It certainly has been great for my career. And because of Jim Womack and his generosity early on in his career and throughout the time that we shared with him here at Texas A&M University, I've clearly benefited from his work and his influence, as many of us have been. And so with that, I'll leave you and take any questions. If I'm not over. Or are we going to take questions? Steve.
I have a general question. Had it for 50 years. What is the functional driver of conserved synteny in small regions of genes that stay together forever, or in larger regions like the ones you talked about here? You had to come up with a list of selected pressures. What would be the top 5?
Cis-interacting regulatory, Sorry, so Steve's question was, what is the basis, genetic basis for extreme conservation of synteny, either locally or across deep time? Well, I tried to touch on some of the different aspects that could be explaining conserved synteny across a large chromosome. But I think there's pretty good evidence that local cis-regulatory effects are constraining most of those shorter regions, right? And we even know that there are multi-megabase regions where enhancers might be extremely distant from their promoters of specific genes, and that disrupting those can actually result in, infertile or deceased rodent models. So I think there's a pretty good understanding that these cis-regulatory interactions are important. I think likely they have to do with TAD structure, or this topologically associated domains. So I think as we disrupt TADs, there's a very interesting literature about what happens when you disrupt these TAD-like structures, which obviously are cis-interacting elements between both the regulatory apparatus for a gene, and if those TAD boundaries start to bleed into each other, you get these remarkable disease phenotypes. So there's some very interesting literature there. So I think that's, in a nutshell, that's what I would say are the likely causes. Richard.
Somewhat related, have you got any comments about the pseudo-autosomal region conservation? Possible implications for conservation of the gene order?
That's a great question. So Terry Roudset, my colleague here, is probably the best suited to answer that question. We'll see if I'll pass. So or with her here in the audience, I'm sure. So the pseudoautosomal region varies quite a bit in size. So in mice, there's actually a fairly short pseudoautosomal region. There's been some implications that actually variation in its size drives, I think, hybrid sterility between some strains of mice. But for the most part, most other placental mammals have a fairly long pseudoautosomal region, and it's fairly stable. It's often anywhere from, I think, as little as 3.5 megabases in humans and horses to as much as 6.5 to 7 or 8 megabases in other placental mammals. So it's remained fairly conserved, and I think it's probably one of the more stable parts of the X chromosome, at least in terms of its size. It also, as you'd expect, has one of the highest recombination rates on the X chromosome. And so consistently we see as part of that undulating landscape, the pseudoautosomal region has this elevated rate. But in terms of what role divergence there might play a role, I think, we don't have a lot of great examples, but it does seem to be, at least for the farm animals and the species that have been looked at in greatest detail, it seems to be generally conserved in terms of its size and that border. Whether there's a functional constraint driving that border and its movement is unknown. But I know Dr. Roudset has lots of thoughts about that. So, yeah, good question. Leif.
Interesting data with the X chromosome, also with the supergene. So the classical feature of the supergene is that you have elevated genetic divergence between haplotypes.
Right.
The high DXY. So do we have that?
We do. So actually, I didn't show the data, but Nicole's shown this was between species phylogenomic data, but we've actually looked at rabbit interspecies. I'm sorry? Right, so this is between species, but within subspecies of rabbits and pigs and cats, we see elevated divergence within the XLRD, both at FST and DXY. So.
We have assembled rabbits, two subspecies of rabbits,
Right.
with the same strategy as you and I. So that's the way to analyze those data. So we should.
Yeah, so we didn't, this was published data by Carnero et al., right? So I believe you were co-author on that paper, right? Carnero et al. On the, yeah, okay. That's what I thought. So yeah, so there we do see, and that paper I believe showed there was elevated DXY. And so we do see evidence for this, like I said, in the few intraspecific comparisons we looked at. So yeah.
This was based on the domestic assembly. Now we have assembled the wild subspecies, the two Napios and HGA. Just analyzing those data, including hybrids from the hybrid zone.
We should definitely talk. Okay, that's exciting. Steve.
Because this is a look back in some ways and a historical comment on the evolution of thought, I might mention it, and I don't know what I would expand on it, about the idea of developing haplotype-specific genome assemblies, which you have demonstrated using the trio binning effect with cats and cattle. But you can't do that in every species, and there's another way, which Evan Eichler of informatics people use, which is to take advantage of a discovery made when Bill was in short pants, okay? And that was the discovery of the hydatidiform mole. The hydatidiform mole, most of you haven't heard of it, is a rare tumor that occurs in a pregnant woman, which basically she thinks she's pregnant, and then at about month number 4, she discovers that she doesn't have a fetus, she has a tumor. And that tumor was discovered by Rabashi Surdi in Pittsburgh, and we worked together in the 70s before anybody ever heard either. And discovered that the tumor was homozygous at every single locus, every polymorphic neural locus that we knew about. And it was equivalent to an inbreed clone of the father of the baby. So the nucleus, the female gets inactivated, and the male, in the hydatidiform mole, the genotype is completely homozygous. And this is what Evan Eichler and the informatics team used for the great apes to do the sequencing, because they couldn't make an interspecies hybrid between a human and a chimpanzee, or if they did, they didn't tell you about it. But the truth is, there are discoveries that occur in different ways, such as the Engelmann's example you use, that just happened to show up. And that happened to show up years ago, and it was used. So keep your eyes open and keep working on things you don't think will amount to anything, and then capture it with a brain like Bill's that goes through the whole literature and picks these things out. So you might want to say a little more about the moles.
Yeah, so to my knowledge, I mean, the mole was obviously a unique example and a unique resource for the Human Genome Project because it essentially created an effectively haploid system. I don't know of the frequency of those or if they've ever been noted before in other mammals, but the newer technologies with the combination of highly accurate, high fidelity reads from PacBio combined with ultra long reads, if you have a trio, even if it's not a divergent interspecies hybrid, as long as you have a trio and there's some good interspecific or intraspecific variation between those parents, you can now get very close to T to T, if not T to T, using the same approaches that basically they developed for the Human Genome T to T project. They've now packaged it into a convenient program called Virco, and that will take essentially those data. If you have the parental data, even better, and for the most part, spit out largely, or well, maybe many of the chromosomes at T to T, and then you have to sort of grapple with the tangles and the other problems, the problematic parts of the assembly to get to T to T. But the technology is moving fast. There's a ruminant T to T consortium. Many of the members here are involved. We're doing our own T to T consortium in the lab in cats, and many other groups are following, and I think the next decade is just going to be an incredible explosion of new discoveries in science because of this dark matter of the genome we've never been able to look at before. Yeah, David.
Yeah. So Bill, how does the fifth base relate to all of this, the methylcytosine? You know, because you know, we typically think about, you know, the primary sequence reads, but when you think about the functional genome, you think about even, you know, highly consistent differences between species. Methylcytosines can have a lot of similarity, but a lot of differences.
Right.
And how does that play into any of the sort of evolutionary aspects?
Yeah, so I mean, I think I showed the one example. One of the beauties of Oxford nanopore reads and some of these longer reads is you can actually obtain methylation data. And so they've been able to note, obviously on the X chromosome in humans, they could show that obviously this DXC4 region and other regions have these unique methylation patterns. In terms of a comparative perspective, I don't know that how rapidly these do evolve between species. That's certainly something we can start looking at with our own data. It's not something we've assessed. I'm not an epigeneticist, so I won't go on a limb, but I did manage to show that example where just the differential methylation was a likely component, differential methylation due to those copy number mediated effects, producing at least what we suspect hybrid sterility, or producing that hybrid sterility phenotype. It's obviously responsible for FSHD in humans. So I know there's other methylation-induced effects due to copy number variation changes. So I think we're going to be seeing a lot more of this. It's just the tip of the iceberg. Yeah. Yes.
So I love that you're getting asked defense questions. This is highly entertaining to me.
I'm done, just kidding.
So I guess this is more of a comment maybe towards the last question there, which is I am an epigeneticist and it's taken me more than a decade, but we're finally able to get comparative methylation maps. And I would just give a word of warning to using any methylation calls off of long read sequencing. So yeah, we can talk about that later.
There you go. Right, anything else? Yeah, someone in the back. Oh, I know who that is.
Yes.
Hi.
Well, thank you so much for a wonderful talk, and it's very inspirational for people who are now working on mammals. I work on insects, and so based on all the insights that you have gathered over decades, do you have any sort of recommendation or suggestion going beyond mammals for other diverse organisms and how comparative genomics that can address some of these questions by expanding the diversity?
Right. So I think one question that came up in a recent talk that I gave was this idea of convergent evolution, right? And so often we see these remarkable changes. For example, in this particular case where I showed the recombination rate maps between human, pig, and cat, the question is, well, is that convergent evolution or is that shared ancestry? And we often make these cases about convergent evolution or shared ancestry in the inadequate phylogenetic sampling. So as a phylogeneticist, I would always encourage to sample deeply, be cautious of cases of convergent evolution. But even now, I realize your genomes are extremely large, but I think still within reach. The costs are coming down. I mean, I don't know how much it costs to sequence one of your grasshopper genomes, but it's a lot more money than it costs to sequence a cat. But I think it's within reason, and I think completeness is essential. I think we have to now consider that the new standard, getting as close to T, because you just don't know what you don't know. And I didn't want to use this platform or this time to talk about the many examples in the literature where early on in comparative genomics, he would say, oh, humans have more of this compared to mouse, or this species has more of this, when there just weren't comparing equivalent genomes. And so we just have to be real cautious about that. So having that standard for T and dense phylogenetic sampling, I think, is really important for comparative genomics.
Hey, Bill, I've got a question for you too.
Okay.
And it's, I think, related to genome size, but I think it's from your next slide. You're talking about introgression was the least, this one, the least on the X chromosome, and there's some chromosomes that had a lot more introgression. How does that relate to chromosome size or chromosome length? Because there's a lot of literature on longer chromosomes exhibiting much larger recombination deserts and short chromosomes undergoing recombination. So is that what you saw here in these studies?
Yeah, actually. So I don't know if these are our best examples, the way this is depicted, but indeed, yes. So we've looked at recombination maps from across many different species. So if you look at an aardvark, for example, has 7 pairs of chromosomes. Its largest chromosome is over 750 million base pairs. But the recombination maps that were generated in my lab, Nicole and Isabella, are just beautiful. They show these huge smiles, high at the ends, troughs in the middle. Those are unusual. And what I didn't talk about is that actually we're trying to discern, are there any other exceptions? Are there other parts of the autosomes that are consistently hot or consistently cold? And so I would just say stay tuned. But in general, yes, we see this pattern where on larger autosomes, we tend to see higher introgression at the ends, depletion in the center, but not quite to the same extent that we see on the X chromosome. But we do see that, and it's likely that larger autosomes also harbor genes because of that reduced recombination and the strength and effect of background selection.
Thank you.
I think I'm probably out of time.
Yeah.
All right. Thanks, everyone.
Great talk. I hate to interrupt this terrific discussion, but we need to move along. And there will be time through the day to talk to Bill about his research.